Hi everyone, my name is Lisa Tostanoski. I'm a graduate student in Professor Chris Jewell's lab at the University of Maryland. And I'd first like to thank the Lemelson Foundation and the Lemelson MIT program for the opportunity to tell you a little bit about what I've been working on for my PhD research. So I thought I'd start by telling you a little bit about how I got involved in the CureIt category and inspired to work on healthcare research. So this is a picture from my senior swim meet in college, and you might notice that instead of holding a cap and goggles and getting ready for my last race, I'm holding a sling from shoulder surgery. And so my experience as a patient, learning from my doctors how scientists, engineers, and clinicians are coming together to design new strategies that might promote healing, really inspired me to pursue biomedical engineering in college and now in graduate school. And so the title of the project I've been working on is Local Reprogramming of Lymph Nodes with Biomaterials to Combat Autoimmune Disease. And so that might spark some questions like, what's an autoimmune disorder, and what's a lymph node, and why biomaterials? And why might we want to combine some of these ideas into a single project? And what kinds of challenges in the healthcare field might we be able to tackle with this idea? So hopefully I'll tell you a little bit about that today. So just some background on how the immune system works. So typically we think of when you get a cut or a cold, immune cells develop that should be able to go out, recognize, attack, and destroy that pathogen to keep us healthy. And that process of attacking a specific molecule is coordinated by our lymph nodes. So these are the tissues that swell up when we get sick because that's where our immune cells mature and are essentially manufactured. So here in blue and white, we can see some of those immune cells in a histology image of a lymph node. And unfortunately, during autoimmune disease, our lymph nodes and other tissues are churning out cells that are malfunctioning. So instead, they're recognizing our own cells and tissues as foreign and attacking. So an example of an autoimmune disease is multiple sclerosis. So this is a disorder in which myelin, which is the protective coating that insulates our brain and spinal cord, is attacked by cells of our immune system. And the way that's treated currently is to use broad immunosuppressive drugs. And these have been incredibly beneficial for patients because it can help control those cells that are attacking myelin. But unfortunately, they don't specifically target those cells that are malfunctioning. So they can leave patients immunocompromised and susceptible to infections and other side effects. And if we think about it a little bit from an engineering perspective and a delivery problem, when these medicines are given through kind of conventional injection routes, like an intravenous injection or in the muscle in your arm, we don't have control over where they go in the body. And so that can expose all of our tissues to broad immunosuppressive drugs. And another challenge is these therapies aren't curative, so patients have to receive frequent, lifelong injections, so something like a daily injection regimen or a monthly infusion in your doctor's office. And so there's a lot of interest in the field, certainly not just in our lab, in whether we might be able to design experimental therapies that could generate more specific control of the disease. So can we teach our body not to attack myelin, but leave healthy, protective immune function intact? And so one way that we might approach this is to bring engineering to the problem. And hopefully we can target something that might be fewer doses, be specific to myelin, and potentially even be curative for these patients. And so just to talk a little bit about how the engineering might come in our lab we work with biomaterials. So you can imagine if you want to deliver something like DNA or RNA, a peptide, a protein, or a drug, you can incorporate these into small assemblies like nanoparticles, microparticles, lipid vesicles, or implantable devices. And some of the advantages of these approaches that have been demonstrated by the field are that you might be able to co-deliver multiple medicines or signals at the same time. You might be able to get them to go interact with certain kinds of cells that you want to target, or get them to go to a particular kind of tissue. And so in my project, we're hoping to take advantage of some of these properties of biomaterials to co-deliver two signals. And the first one is myelin, that molecule that's attacked in MS. And we're aiming to co-deliver that with a drug that can teach the immune system not to attack or to calm down. And hopefully together, when cells see these signals, they can learn to recognize myelin, but rather than attack it, actually protect it from being attacked by our immune system. And so the way we approach this is to program the combinations of signals in lymph nodes, those tissues that coordinate our immune system. And so we have this cartoon in the lab that we like to kind of explain the approach. So in MS, we have these cells I mentioned that are shown here in red that recognize and attack myelin, that protective coating around our neurons. But these cells actually develop in our lymph nodes, the command centers of our immune system. So we have cells that recognize myelin that come in to what we like to think of as a classroom environment. So they interact with other kinds of immune cells and essentially learn bad habits, where they're taught to become inflammatory and learn to attack myelin. These cells then travel out of the lymph node to the brain, where they can have these effects where they damage the myelin sheath. And that can interfere with how the brain communicates with the rest of the body. And so the goal of my project was to apply this biomaterial strategy to reprogram how these cells learn in this classroom environment. So depicted in these blue spheres are our biomaterial particles and encapsulate those two signals I mentioned, myelin with a drug that can hopefully change how these cells learn and develop, instead becoming cells that recognize myelin but are able to travel out of the lymph node and actually protect it from attack rather than driving disease further. And so what we're really excited about is hopefully this might have implications for some of the 2.3 million patients worldwide that are affected by MS, as well as some of the patients that are affected by other diseases. But unfortunately, before we can think about getting this into a human patient, there's a lot of really important steps to understand the characteristics of those materials and maybe use some preclinical models to understand whether this idea might work. And so I'll just show you an example of a model that we work with in our lab. So we work in a mouse model of MS. So this is a typical mouse with the disease. So you can see she's alert and able to move around her cage, but her tail and hind limbs are paralyzed. And that's because this mouse has immune cells that have learned to recognize and attack myelin. And the effects of those cells is that they damage how the brain can communicate with the rest of the body for motor function. And so just after I captured this video, I gave this mouse a single dose of our particles directly in the lymph node. And this is the same mouse 2 weeks later. We can see a pretty exciting difference in level of disease. We've actually reversed paralysis and restored hind limb function, so she can stand up on her hind limbs and run around again. And so what I've been working on in my PhD is to try to understand the mechanism of how this is happening. Are we really controlling those myelin-specific reactions, or does this have any side effects? And I've also been really fortunate to work with great clinical collaborators on understanding whether this might be a platform that we could apply to other diseases, for example, transplant rejection. And so just to wrap up, I just want to thank people that made this research possible, like Tomas and Courtney mentioned, finding great mentors, including my PhD advisor, Chris Jewell, and our clinical collaborators, Dr. Jonathan Bromberg and Dr. Walter Royal. And again, I'd like to thank the Lemelson MIT program and our lab's funding sources, particularly the NSF Graduate Research Fellowship Program. And I'd be happy to take any questions.
Okay, do we have any questions? We have a couple minutes right here in the center.
I'm sorry?
We have a microphone if you can just pass it down. Could you stand up too when you ask?
Have you got a name for your product yet?
A name for the project?
Yeah.
So we've mostly been calling it local reprogramming of lymph nodes. Yeah, I don't know if that's a clever name for it.
Pass it over.
Do you think this would be able to work with the type of autoimmune where you become allergic to pretty much everything?
So,
because my stepmom has that. And I was like, she's pretty much
that.
allergic to everything.
Yeah, I'm sorry to hear that. So the materials we make, like I mentioned, they incorporate myelin peptide with one of these drugs that can hopefully reprogram how the body responds specifically to myelin. And we hope this will be like a modular platform where we could change the myelin out for another kind of molecule that might drive another disease. So we're working with clinical collaborators now to see if we can use this strategy for organ transplant. So can we change the myelin out for something that's attacked in a transplant rejection? And that's a great idea that we could think about changing that for something like type 1 diabetes or allergies or some other kind of application.
Also, what happens if you're allergic to like the basic things that are supposed to deliver the thing?
Yeah, that's a great question. So we use a polymer carrier called PLGA, which is one of the most common biocompatible carriers. But you're definitely right, we run particle controls to make sure that the carrier doesn't have any effects. But that's a great consideration that we could think about whether we might want to change out that polymer or maybe even try to use a strategy that could eliminate the carrier but capture some of these features of biomaterials.
Okay, do we have time for one more question?
You said that with that one mouse, the myelin grew back. Does it grow back very readily in humans? Is this a thing that could like be restorative pretty readily?
Yeah, so that's a great question that we're trying to study in the mice now. So we see a reversal of symptoms, and what happens in MS is we have cells that directly damage the myelin, and they also damage the cells that are designed to reproduce and regenerate myelin if any damage occurs in regular life or in disease. And so we're trying to understand in the models now, are we actually protecting those cells that should regenerate myelin, maybe even getting new myelination? And then we'd have to think about whether if we see that in the mice, might that have translational potential for humans? But yeah, that's a great question.
All right, thank you. We're out of time. So next up, we have Katie.